So now we're going to focus on the central dogma. The central dogma has to do with the fact that we have DNA that contains the instructions to make a messenger RNA transcript, which is then read in order to be turned into a sequence of amino acids that can then be folded into a protein. So if we have a strand of DNA, now DNA is double-stranded, but typically when we're talking about transcription, we only make a messenger RNA copy of one of the strands. DNA has regions known as genes. And the gene contains the sequence of amino acids or sequence of nucleotides that is going to be able to give us our amino acid sequence. So a gene codes for a protein. Now DNA is really long and winding, and we have to know where the start of a gene is and where the end of a gene is. So in order to figure this out, upstream of the gene, we usually have a promoter region. And then at the end of the gene, we have a termination sequence. So when we make a copy of the gene, we copy a little bit of the promoter region, and then we copy a little bit of the termination sequence. And when we make this copy, we make a copy called messenger RNA. So let's get into the process of transcription. So transcription is the process of making a messenger RNA copy of a gene of interest. So it's the process of making a messenger RNA copy of a gene of interest. So if we have our double-stranded DNA here, an invisible region in here is the promoter for the gene. And then another invisible region is going to be the gene itself. And then there's going to be a termination sequence. So what's going to happen is RNA primase, or polymerase, binds to sigma factor to form a holoenzyme, and then they bind to the promoter together. So there's this enzyme known as RNA polymerase. And then there's this little tiny sigma factor. And these two are going to come together, and then they're going to bind to the promoter. So the function of the sigma factor is really to guide RNA polymerase to the promoter. As soon as RNA polymerase is by the promoter, it starts to unwind and open up the strands of DNA. So we just talked about DNA replication and how helicase opens up the strands of DNA. Well, when we're talking about transcription, RNA polymerase opens up the strands of DNA. This completes the process of initiation of transcription. The second step of transcription is elongation. So what happens during elongation is now RNA polymerase has opened up the double-stranded DNA. Sigma factor falls off, and RNA polymerase, which I'll abbreviate RNA pol, begins to make a messenger RNA copy of the DNA. So RNA polymerase will go down this strand, and as it goes down the strand, it will copy the DNA. Now the promoter was here. The gene of interest is this region right here. And then at the end of the gene is a termination sequence. So if we look at our open DNA, RNA polymerase is now at the end here. We have made our messenger RNA copy. And as soon as RNA polymerase hits this termination sequence, it's going to bump into this small factor known as Rho. So when RNA polymerase hits Rho, this entire thing falls apart and the messenger RNA will float away to go on to the process of translation. So remember, if we're talking about the regions of the messenger RNA here, we had the promoter region here, we had the gene of interest here, then we had this termination sequence. And inside of the termination sequence sits Rho, and as soon as RNA polymerase bumps into Rho, this whole thing dissociates, and the messenger RNA that was produced goes off to a ribosome to be translated. So now we get to translation. And when we talk about translation, we have to talk a little bit about the ribosome. The ribosome has two subunits. And these subunits are normally independent of each other. They only come together when messenger RNA is present. So let me actually delete that. We'll start out with the small subunit. Now when messenger RNA binds to the small subunit, that stimulates the large subunit of the ribosome to also bind. Now, through the process of translation, the ribosome reads the code of the messenger RNA and converts it to a sequence of amino acids. So translation is the process of reading the messenger RNA and translating it into a sequence of amino acids. This large subunit on the ribosome is going to have three major sites: the A site, the P site, and the E site in reverse order. So we actually have E over here, P here, and A here. The A site of the ribosome, also known as aminoacyl site, this is the site of incoming tRNAs. So this is where the transfer RNA anticodon is going to measure up with the codon of the messenger RNA. And I'll talk more about that in just a little bit. The P site for polypeptide is where the growing chain of amino acids is going to stay. So as this protein is being assembled, it's assembled in the P site. And then the E site for empty or exit is where the empty tRNA exits the ribosome. So it's going to leave and go get a new amino acid to be able to bring into the A site. So messenger RNA isn't just a string. If we look at messenger RNA closely, messenger RNA is a series of bases. So if we have A, C, U, G, A, U, G, C. Now the reason why I'm writing U's instead of T's is because this is RNA. If we go back to this picture for a minute, all of these lines represent bases. So the original sequence up here was a DNA sequence. So we had A, T, C, G. And when we went to make our messenger RNA, we made a complementary sequence. So A would bond with U, T would bond with A, C would bond with G, and G would bond with C. And this is how we copied the DNA. So our DNA sequence was first, and we copied it to make a complementary sequence of messenger RNA. Well, now we're going to read this complementary sequence of messenger RNA and turn it into a chain of amino acids. Messenger RNA is read in series of 3 bases. So every 3 bases marks a codon. So codon is 3 bases on messenger RNA that codes for 1 amino acid. So this is the code here. UUU codes for the amino acid phenylalanine. So if our messenger RNA said UUU, we would incorporate the phenylalanine amino acid. If the next 3 letters were CCU, we would incorporate the amino acid proline. If the next 3 letters were CAA, we would incorporate the amino acid glutamine, and so forth. So we actually read the codon, or the sequence of 3 amino acids on the ribosome, in order to figure out what the next amino acid is going to be incorporated into the chain. But we have to start somewhere. Now you'll notice here there's a start codon, and then there's 3 stop codons. A start codon is necessary to start translation. So this is how we know what the start of the protein is. So if this is our messenger RNA, and this messenger RNA is going through the ribosome, so we have A, C, U, G, A, U, G, C, and so forth. It's going to ignore everything until it gets to the point where it says AUG. And when AUG is aligned underneath the A site, it's going to start translation. Meanwhile, this thing is going through the ribosome in this direction. So it's going to keep traveling through until it gets to where it sees AUG. AUG is our start codon, and this is going to start translation. So here's a picture that shows the ribosome subunits. So the subunits are separated. Once messenger RNA binds to the small subunit, the large subunit will also bind. Once bound, now we're going to start making a protein. So messenger RNA will be ignored until it gets to a start codon. And the start codon in this location, again, would have to be an AUG. So this first amino acid that is always incorporated is methionine, because it has to start with the AUG start codon. Now these things called transfer RNAs, tRNA, carries amino acids into the ribosome. And the transfer RNA has an anticodon. And that's 3 bases on tRNA that will bind to a codon of messenger RNA. So if I just diagram that real quickly, we have our ribosome here. We have our messenger RNA in here. We have AUG under the A site of the ribosome. Our transfer RNA actually looks like the letter T. It carries in an amino acid, and it has these 3 bases down here. Now this transfer RNA will migrate over to the ribosome, and it'll enter into the ribosome, carrying its amino acid. And this anticodon, UAC, will bond complementarily to the codon, AUG, of messenger RNA. So now these are bonded. You have the anticodon of tRNA, you have the codon of mRNA, and now this amino acid will be incorporated into the protein. Well, this whole thing is going to shift. And when it shifts, this gets brought over to this site. Our messenger RNA shifts as well. So our AUG is now under the P site, and a new sequence is under the A site. So now a new transfer RNA carrying whatever would be complementary to here would be brought into the ribosome. So its anticodon would have to bond, so we'd have to have UUG on our anticodon. And this one carries a different amino acid. So if we look at AAC in the table here, AAC means that the next amino acid to be incorporated into our chain would be asparagine. So this green amino acid that I drew here would be asparagine. So now continuing on with translation, we're going to move this thing one more time. So now this one is in here empty because they leave their amino acids in the P site. So our green amino acid's here now, and then our red amino acid is attached to it there. Now we have a different codon here, so we would bring in a new amino acid. And this keeps shifting down. This one will exit the ribosome. This will shift over and over and over, and this growing chain of amino acids is going to stay in that site. So this is what is pictured here. So you can see as it shifts, it'll grow into this chain of amino acids. There are some major differences between prokaryotic and eukaryotic transcription and translation. If you think about it, prokaryotic cells do not have a nucleus. They just have some genetic material. Eukaryotic cells do have a nucleus. So their DNA is inside their nucleus. That means that transcription can happen here, and translation has to happen outside of the nucleus. But because in prokaryotes, they don't have a nucleus, While their messenger RNA is being produced, a ribosome can immediately bind to it and start producing protein right away. Prokaryotes, one RNA polymerase makes all types of RNA, while in eukaryotes it requires multiple RNA polymerases. In prokaryotes, transcription is initiated using sigma factor. In eukaryotes, you need a lot of different transcription factors. The promoter is at the start of the gene in prokaryotes, while there's usually a TATA box and a bunch of other things upstream of the promoter in eukaryotes. Prokaryotes, one promoter can control the expression of multiple genes incorporated into an operon. So we'll discuss the operon in detail next. In eukaryotes, one promoter, one gene, one protein. So you make a messenger RNA, you make one protein from it, and then you immediately degrade it. Introns and exons we'll talk about in the next slide. Prokaryotes do not have introns, eukaryotes do. And then with prokaryotes, messenger RNA again can be translated immediately. In eukaryotes, there has to be some editing involved before the messenger RNA can be translated. So what is an intron? If we have a sequence of DNA, there's the coding regions, and then there's non-coding. Non-coding regions are introns. While the coding regions are exons. These introns have to be removed prior to translation, or you're going to end up translating the wrong protein. So what we do is we take this segment of DNA, we cut out the introns through a splicing process, and then we glue together the remaining pieces. And the remaining edited mRNA is much shorter in length. We're not sure what's going on with these introns and why they're here, but if you remember the slide that I went through on the first part of this lecture, only 2% of our genome actually codes for protein. So what is the rest of it doing? Well, like 80% of E. coli's genome codes for protein. We need to do more research on these introns to see what they're all involved in. There's a couple books published recently that talk about what these introns do and how they're associated with our intelligence and all sorts of other stuff. So that's kind of research for the future.